Hello and welcome to my presentation. My name is Sebastian Bull. I work as a research scientist at the University of Texas Health Science Center in San Antonio, Texas, where I study the humoral immune response to infection with the malaria-causing Plasmodium parasites. In my free time, however, I do scientific cat research at Cowboy Cat Ranch. I'm delighted that I can share our latest research findings on cat-attracting plants with you. I owe a lot to our collaborators in Australia, Gavin Flamatti and Adrian Scafidi. They synthesized most of the single compounds that we used, and they performed the chemical analysis of the cat-attracting plants. Furthermore, I want to thank my wife, Feline for her assistance with the bioassays and for allowing me to use our home as a USDA-registered research facility for the past several years. When talking about cat-attracting plants, most people will think of catnip. The response to catnip appears euphoric and blissful, and it is quite bizarre that it has been so poorly studied. Prior to our studies, we knew close to nothing about the catnip response, and as of today, we still know only very little. We, for example, still don't know why cats and only cats respond to this plant, and we also still don't know which olfactory receptors are involved. After we had adopted our cats from the local shelter, we decided to keep them inside with an outdoor enclosure for many good reasons. Therefore, we felt obliged to enrich their indoor environment. When we gave them catnip, dried or fresh, there was absolutely no interest in the plant. Ollie seemed to enjoy sitting in the pot of the catnip plant, though. The internet is full of anecdotes about other plants with effects claimed to be similar to the effect of catnip. Of these plants, silvervine had been described in literature, but bioassays with cats have only been done with catnip. Therefore, we decided to test what the response frequency was for several of these other plants. We offered 100 shelter cats catnip, silvervine, Tatarian honeysuckle, and valerian. We were curious to see if cats who did not respond to catnip would respond to some of the other plants. Each plant was available on at least 3 different days, about 1 hour each day. A positive response was defined as engagement with the plant material that was not seen in response to the negative control. Typically, head, chin, and cheek rubbing, rolling over, raking, or body kicking, and biting were seen. We found that about 70% of the cats responded to catnip. And this percentage is in strong agreement with the results previously published 40 to 50 years ago. Slightly more cats responded to silvervine, and about 50% of the cats responded to Tatarian honeysuckle and valerian. Now, what about cats like Ollie who didn't respond to catnip? So of the 100 cats, just over 30 didn't like catnip. Of those cats, many responded to silvervine. About 80% of the cats who did not like catnip responded to at least one of the other 3 cat-attracting plants. And overall, almost 95% of the cats responded to at least one of the 4 plants tested, demonstrating the usefulness of them in environmental enrichment for cats. The take-home message here is that cats who do not like catnip may love other plants. Now, the outcome of the previous study was rather black and white, response or no response. We did not look at the actual behavior of the cats. With our latest study, of which the results were published only 2 months ago in BMC Biology, we wanted to assess possible differences in behavior. Differences in response between the plants, but also differences between the cats. In this study, we included a 5th plant called Indian nettle. 6 cats at Cowboy Cat Ranch, again our home, were offered these cat-attracting plants together with a negative control, tea leaves. Each plant was available to them for 100 hours, spread out over a 5-week period. All 5 plants were tested within a year. First, we looked at the duration of the response, and we compared this between plants and between cats. Here you see that all the cats responded to catnip. Each white dot shows the total response time per cat. As you can see, the response duration to the other cat-attracting plants was highly similar, meaning that catnip is not king or queen. It seemed that cats preferred one cat-attracting plant over some of the others. Harvey, for example, responded significantly longer to silvervine, whereas Ollie responded longer to Tatarian honeysuckle. When comparing response duration between cats, we found big differences between them. Here, the total response duration for each plant is shown as a white dot, and you can see that Ollie responded much longer to the plants than some of the other cats did. Next, we studied the behavior of each cat in great detail. We created an ethogram and a video ethogram, which is available online, specific for responses to cat-attracting plants. We found that the behavior in response to cat-attracting plants differs enormously between cats. Some cats mostly displayed head rubbing while in a sitting position. Others showed more dynamic responses while lying on their side, where the object was actively held between their front legs, rubbed, bitten, and kicked with the hind legs. One special observation, as you can see on the right, was rippling of the back in response to smelling these plants. This rippling of the back should not be confused with feline hyperesthesia syndrome. After testing more than 100 cats, we have not seen a single case of aggression resulting from smelling these plants. Playfulness should not be confused with aggression. Cats should not be petted during their response. Aggression is always possible when there is scarcity. When there are 3 cats, for example, it's best to have 4 toys with cat-attracting plants available. Many of these differences in behavior between the cats were statistically significant. Please note that rippling of the back has been overlooked in the past, but is seen in a substantial portion of domestic cats, 3 out of 6 in this study. The take-home message here is that the behavior in response to cat-attracting plants differs a lot between cats, but not between plants. Before we started studying the catnip response, very little was known about it. What was known is the molecule responsible for the reaction to catnip, or Nepeta cataria, which is nepetalactone. However, we now know that cats also respond to several other plants, and we had shown that these plants do not contain nepetalactone. Silvervine contains molecules with a structure similar or somewhat similar to nepetalactone, but bioassays with cats were lacking. We decided to test about a dozen of single compounds. The molecules were dissolved in diethyl ether, which was used as a negative control. During both testing and analysis, I did not know which sample was which compound. Each compound was tested on at least 2 different days. The goal here was to see which compounds were able to elicit the catnip response. As expected, we did not see a response to the negative controls, which were available for more than 170 hours. While these molecules have names, these names do not give any information about their structure. Therefore, I'm also showing the structure of these molecules. As expected, nepetalactone and apinepetalactone both were able to elicit the catnip response, and so were all other molecules that were highly similar in structure to nepetalactone. These 6 molecules are all called type 2 lactones based on the location of their carbonyl group. The type 1 lactones, with a different position of the carbonyl group, also elicited the catnip response. Dihydroactidiniolide is the only single compound that we tested which is not exclusively synthesized by plants and insects. This molecule was also identified in glandular excretions and the urine of the red fox. Although the response duration to this compound seemed to be shorter compared to some of the other cats, compared to some of the other cat-attracting volatiles, the observed behavior to dihydroactidiniolide was near identical to the behavior seen in response to nepetalactone or catnip. Lots of head, chin, or cheek rubbing, some biting, raking, rolling on the side, and head shaking. Finally, actinidin. This single compound is structurally the most different from nepetalactone because it contains a nitrogen atom and lacks the carbonyl group. The first 9 molecules can collectively be referred to as lactones, whereas actinidin is a pyridine. The response to actinidin was quite different compared to the other compounds, in that the 3 siblings did not respond to it at all, while there was one cat who responded a lot to it. Out of the 5 cats who participated in this experiment, these three siblings, cats N, O, and V, responded the longest to the lactones. But when looking at actinidin, you can see an almost perfectly inverted image. To replicate what we observed, actinidin was tested on 3 additional days, and the results observed were the same. We also found that the behavior in response to actinidin was somewhat different, since licking the object with actinidin was observed, which was not seen in response to the other compounds or catnip. While our manuscript was in preparation last year, an article about cats and silvervine was published, and in their supplementary online material, we found some interesting raw data that surprisingly had not been discussed in their article. 11 of 12 cats responded to several lactones, but not to actinidin. However, there was one cat who did respond to actinidin, and this cat seemed to love this molecule more than any of the other cat-attracting volatiles. These findings are in striking agreement with our results. We also measured the time to first response to assess how strongly the cats were attracted to the molecules. In line with expectations based on the previous results, cat A was stronger attracted to actinidin than to the other compounds. And for the 3 siblings, we observed the opposite. The take-home message here is that not just napidolactone, but a dozen of other molecules are able to elicit the catnip response, and that the disparity between the lactones and actinidin suggests that more than one olfactory receptor may bind these cat-attracting molecules. We next did experiments to learn if cats habituate to cat-attracting plants. We did this by offering one of the plants for 10 days in a row. As you can see, habituation occurs for all cat-attracting plants that we tested: catnip, silvervine, and Tatarian honeysuckle. Therefore, it is recommended to not have toys with cat-attracting plants available continuously. It's probably best to give these plants to cats no more than a couple of days per week. These experiments revealed something quite interesting. Remember that Ollie responded longer to Tatarian honeysuckle than to the other plants. When we offered Tatarian honeysuckle after the cats were habituated to catnip and silvervine, we noticed that Ollie responded a lot to Tatarian honeysuckle. In these graphs, the black dot is Ollie. His response duration was even longer than the initial response to catnip or silvervine. This was not observed when he was exposed to catnip after habituation to Tatarian honeysuckle. These findings suggest the presence of a molecule in Tatarian honeysuckle that Ollie enjoys and which is not present in catnip or silvervine. However, when we look at the results of the chemical analysis of these plants, we see that none of the compounds present in Tatarian honeysuckle, THS, are absent or present at lower levels in silvervine, SV, or catnip, CN. These findings suggest there may be unidentified cat-attracting single compounds present in Tatarian honeysuckle. Interestingly, our 5 cats were part of the 100 cats who participated in our first catnip study that we published in 2016. None of the 5 cats responded to catnip back then. Years later, however, all 5 did. It has been mentioned that kittens don't respond to catnip, so we wondered if the ability to respond to catnip is something that needs to develop over time. We indeed observed that fewer of the younger cats, 6 to 18 months old, responded to catnip than the older cats did. However, 2 of the 5 cats were between 4 and 10 years old when given catnip. Of the 31 cats who did not respond to catnip in that study, our 5 cats were exposed to catnip more frequently and longer than any of the other cats because they were living with us at home. Could this mean that every single cat has the ability to respond to catnip? Did exposure to silvervine have anything to do with this? The answer for now is we do not know. One more important thing, no matter how funny some of these cartoons or pictures on the internet may be, any association of cat-attracting plants with human psychoactive drugs is undesirable and not supported by science. Many cat guardians shy away from giving their cats cat-attracting plants because they feel they are doing something that's bad for their cat, while we all know that these plants are great at stimulating play, increasing activity, preventing boredom, and even improve social interaction interactions between cats. Here are some important differences between human psychoactive drugs, such as cocaine, heroin, or THC in cannabis, and cat-attracting plants. The cat-attracting plants are not even distantly related to the plants used for human drugs. Cat-attracting plants only have an effect when smelled. The active molecules are small and look nothing like the human drugs. The euphoric response ends immediately after removal of the plant tissue, and no withdrawal or dependence has ever been observed. Now, in conclusion, the things to remember are catnip isn't king or queen. Cats who don't like catnip may love other plants. Responses between cats are highly variable. Cat-attracting plants do not cause aggression. Responses can be elicited by at least a dozen molecules. There may be more not yet identified cat-attracting compounds. Actinidin may work through different receptors. All domestic cats can respond to catnip? Cat guardians are not turning their cats into drug addicts. Here's your summary slide. You can pause the presentation here if you wish to take a picture of it. With that, I want to thank you for your attention. If you have any questions or are interested in doing scientific research together, please send an email to meow@cowboycatranch.org.